Okay, so today is the second lecture of the DNA sequencing, and we will really get into the bioinformatics component of it, especially for today and tomorrow. And there will be a couple of major topics regarding the data processing to get the sequence alignment, to get the variant identified, and then how do we clean up the data. From last time, there are different types of genetic variants that we want to identify from the whole genome sequencing or different level of DNA sequencing data. The first one is the SNP, and there are 2 points I want to make. And the first one is if you qualify for that SNP, if only one individual have it, normally it is only called single nucleotide variants. And to qualify as a polymorphism, you better have a significant proportion of population have it. So the second one is the translation and transversion ratio. So we know that the translation is within purine or permitting, and the transversion is between purine and permitting. So if every change is random, that will be 0.5 for the translation transversion ratio. But in reality, it is much bigger than that. For the whole genome sequencing, it will be 2.0 and around that, and for whole exome sequencing, it's anywhere from 2.8 to 3.0. So that is one of the ways that people use to see whether their sequencing result makes sense. The second type is the short insertion deletions, and then there are different levels of repetitive elements, including the microsatellites, and mini satellites and the repeated genes, and the only difference for that is the size of the repetitive elements. And there are large structure variations, including large insertion deletions, duplications, inversions, copy number variations. Some of them can be quite big, but normally to qualify as the big ones, it's normally a one larger than 1 kb or a few kbs. Before we had the next generation sequencing technology, and initially people have the microsatellite markers, and then eventually the SNP arrays start to come along, starting from a low resolution, like 6 SNP markers across the genome, all the way to 1 million, and maybe 2 million or 5 million SNPs. And for the analysis, initially, it is more on the linkage analysis side, but now because of the marker become much more dense, and people are doing the association study on those. But specific topic, talking about association study, the genome-wide association study, which is the GWAS, that has been done for many years and lead to many important scientific discoveries. But one of the assumptions of the GWAS is common disease, common variance assumption. And we have gone through this figure. X is allele frequency, so larger, that means it's common SNPs. Y is the effect size to see whether having that SNP will increase or decrease your disease risk. And we mentioned for here are the ones that are either not life-threatening or too much disease-related phenotypes, or it has been low-hanging fruit that's already been picked. For the GWAS study, because of the way that the technology, we have to use the GWAS arrays. And in order to put any SNP markers on the array, we will have to have a substantial proportion of people has it, so that it qualifies as a common SNP. So in this spectrum, you see that when we go to common SNP, by definition, the effect size of your findings will be small. So that is one of the major issues or challenges of the GWAS data analysis. And for this spectrum, you'll see it's very, very rare SNPs, but once it happens, it has a large disease phenotype. Those are more Mendelian disease that usually go with the family structures. And those are normally the linkage study will be more helpful in this spectrum. But for whole genome sequencing or different level of DNA sequencing, we will have the opportunity to go over all this. Okay, So for the GWAS study, the assumption is common disease, common variants. There are many different limitations. The first one is because of the design of the SNP chips, and these cannot capture rare variants, only focus on common ones. And it really doesn't have the functional information, which means if you identify a genetic marker, SNP, that is associated with a phenotype, that doesn't mean this SNP is causing the phenotype. That only means it's associated with that. So it's more than likely there's another SNP in the same LD regions that is being tagged by this SNP, it's causing that phenotype. And also there are many different levels of heritability, missing heritability. We discussed briefly about the gene-gene interactions. The GWAS study that doesn't take care of it. And the gene-environment interactions, the epigenetic regulations. So those are being missed by the GWAS studies. We talk about experimental considerations for the DNA sequencing, and we'll start from the whole genome sequencing. And the price dropped dramatically, but still not very cheap. It's about a little bit over $1,000 per genome for the research grade. It could be much more expensive for the CLIA or clinical grade. And also that when I say $1,000 per genome, that is normally for germline mutations and with the standard 30 to 40x coverage. If you want to study tumor, which we'll get into a little bit, and then you will need to have much deeper coverage. So if you want to do the whole genome sequencing, that will become much more expensive. For the target sequencing, and that's the second strategy. So if we sequence the whole genome is too expensive, we can only focus on the region we're interested in. And there is a topic we talk about capture selection strategies. It's either a region that you already have and in mind based on the previous linkage or GWAS studies, or you have some prior knowledge, like a cancer study. There is 50 genes I know that is related to this cancer type. You can always use those strategies. Or if you know nothing about the disease, a safer bet will start from the whole exome sequencing, which focus on the exome regions that will become a protein coding genes. And then we talk about different technologies to do the capture, and more specifically for the capture array and the capture in solution technologies. I'm not going to repeat myself about that. So if for this part, we know that for the whole exome sequencing, it's getting cheaper. It's become like a $350 to $600, depends on the coverage you want to get. But half of those costs is actually in the capture side. Only half of them is in the sequencing part. So whether we can redesign our experiment by pulling multiple individuals' DNA together before the capture station, in that case, we'll save a dramatic amount for the overall cost. So that will come to the pooling strategy. And there are some advantage, which is the major advantage is really the cost. And but the challenge for that is the data analysis, and it doesn't give you the resolution for the individual sequencing. But in many of the study designs, it may still help you to identify important stuff by including more in the sample size. And then we talk about the long-read sequencing for PacBio, Nanopore, and the linked rates, which we'll get into a little bit more detail today as well. And the reason that I didn't talk about last time is I didn't have that information last time. And this Wednesday, we had this 10x training, and I learned quite a bit from that. So I put a few slides in this lecture, and I will planning to put more in the next one as well. And then there is a single-cell DNA sequencing. This is not the one that the instrument that we have that can do this experiment. The instrument we have can do the single-cell RNA sequencing. For the DNA sequencing, it becomes much more challenging, and there is different issues associated with that. I'm not an expert, but if you're interested in that, you can feel free to look at the literature. But those are basically the summary of what we talked about last time. Okay, so now let's get to the topic of today and actually in Monday lectures. We know that this is more like the central dogma of the next generation sequencing data analysis. We start from the raw data, and then we get to the sequence alignment, and then do the BAM file. We will get the BAM file. After getting the BAM file, we will do the variant calling, and then eventually get the variant, which is the VCF files. So we talk about alignment in general, but the topic I want to focus in these two lectures, the first one is regarding the sequence alignment, the sequence mapability issue, and whether we can properly address that. And the second one is something called the refined alignment, which we'll get into in a little bit more detail today. And then after doing that, there is two other recalibration we need to make. One is to recalibrate the base quality of the sequencing from the raw sequencing data. Okay, the base quality we mentioned, we have base quality, mapping quality, and variant quality. But for the base quality, and it's reported by the sequencer. Question is, are they accurate? What if they lie? Right, I can easily put a Q30 100% if it's not real, and how do you know that? And whether we have a way to recalibrate that base quality. And then the other one is once we identify the variant calling, whether this is being estimated, the error is being estimated properly. So these are the parts I will talk about next time. And another major topic is going to be the variant identification part. Once you get the BAM file, all the reads are being properly aligned, and then you want to identify the variants. You want to generate the VCF files. But from this spectrum, we need to be careful about our study design, what we are looking at. For diploid genome, which means we have parental and the maternal mutate and alleles, they mix together like humans. And there's one way to do the analysis. If we have multiple individual samples, there's another way to do the analysis. Put the DNA sequencing and the cancer genome, they all have different ways to do the analysis. So we'll get into all these topics in the next lecture as well. Okay, so the first part is the sequence mapability issues. So what are we talking about? Okay, so here is a statement. For one sequence fragment, okay, a DNA, like you sequenced, what is the minimum length, the number of nucleotides that can be uniquely mapped back to the human genome? Any guess?
General consensus is about 20 base pair.
20 base pair? That's a really good guess. And when we design PCR primers, and we are about 20 to 25, and one of the primary reasons is you want to see uniquely mapped to the genome, right? If we put this into a mathematical formula, which never will work, but we will do that anyways. We know the size of the genome is about 3 billion letters, okay? So each nucleotide has 4 choices, A, C, G, and T. If everything occurs randomly, and if we need to have this, right? 4 power n is larger than 3 billion. And in that case, so the minimum number in theory will be 16 nucleotides. We'll at least need that. But we know that the human genome are not random. There are some A and T will occur much more frequent than C and G, and there's a lot of duplications involved or repeats involved as well. So that's why that we pick about 20 bases. But in theory, this is the 16 will be the magic number that you at least need to be beyond that. Otherwise, there's no way you can map your short sequence arrays back to the genome. We have done some of the microRNA data analysis, and some of the samples are microRNA sequencing, right? We know microRNAs are short, 22 bases, and we map them back to the genome, and which is great. We can map it. Most of them map properly. But some of the samples are more degraded than others, and we found that some of the samples and nothing is mapped, and or 80% are not mappable. So what's the reason? When I see the histogram of the sequencing length, and many of them goes to be less than 15 bases, and And in that case, regardless what you do, you cannot properly map your sequences. So, but the point is, this is the number you want to get to at least. In theory, race with 16 base or more can be uniquely mapped back to the human genome. Okay. So human genome, so this is in the case that all the letters in human genome is random, but we know it's not. So human genome is repetitive. Actually, over 52% of the human genome are repetitive DNA. This is kind of, well, if you know it, it's not surprising, but if you don't know it, it's pretty surprising that over 52% of our DNA are repetitive DNA elements. They are defined as the elements that are found in multiple copies, in some cases, thousands of copies or even more. There are many types of repeats, and we mentioned briefly about the tandem repeats, which include mini-satellites and microsatellites. And there are also some of the repeats, like interspersed repeats, like DNA transposons. This will account for about 3% of human genomes. LTR, long-terminal repeats, retrotransposons, 8%. But if you look at this non-LTR retrotransposons, like the sign and the line itself account for 21% of human genome. There are many others as well. So you can see this is specific where is ALU elements are about 300 base long. Chromosomes were hybridized with a probe with ALU elements, and then you can see it occurred so many times in the human genome. Those are the ones will give us a big trouble when we do the sequence analysis, because they are occurring many times in the genome. If we get a sequence for shorter than that, and we really don't know which part it comes from, so that is our issue. But scientifically, these repeats, some of them, I mean, in the past, the people would consider they are not useful. They are just junks. But more and more recent that people start to understand the functions of these repeats. They are not junks. And specifically for ALU. This is something very interesting. It will be introduced in the genome, in the mammalian genome, after the primate is just around the primate is being formed. Okay, and so in the mouse, rat, and those species, we don't have ALU elements. But if you look at humans, chimpanzees, and more primate species, we start to have ALU elements. And then they are so frequent in our genome, and actually our cells start to learn how to use them. And many of the ALU elements, because surprisingly they have these splicing elements in the ALU elements, which make them a very good candidate to generate new exons. And that is one of the reasons why human genome has so much alternative splicing, more alternative splicing than mouse and the previous genomes. So because ALU elements can help us to generate more. And there is a theory that ALU elements is one of the mechanisms that trigger that primate, the brain development are being so advanced comparing to other species. But those are those sad stories. So don't think they're completely junks. They are not. And they have a lot of implications in cancer as well. Some study has shown some of the ALU elements actually become alternative promoter that drive an oncogene to be expressed, and then eventually cause the problem for the patients. Okay. Now coming back to informatics. In that case, okay, if the reference, the human genomes are our genomes are repetitive, short rates, how short is too short? Okay, we know that we cannot go anything less than 16 bases. So you can see this is one of the earlier simulations that people did, just looking at different versions of genome. And if you cannot see from the back, this blue one is HG human genome. And that was done so early, so it's HG18. So we have two versions after that already. So this is at least 10, 15 years ago, okay? But human genome doesn't evolve that quickly, so 15 years ago, that's fine. But the study they have done is they will look at different read lengths, and then they will see that so if my sequencing rate is 35 bases, what are the percentage of the genome I can perfectly identify, I can uniquely map to, okay? So you can see that when this is lower than 20, probably everything is not being uniquely mapped. And then this will start to increase with the length of the sequencing rates increase. And then now you say, why we're doing this? What's the point? But keep in mind, with very early sequencing technology, we only had 25 bases for the Solexa. That's the pre-Illumina, right? Illumina purchased Solexa and make the Illumina today. But that was the very initial version was 26, and then there's quite a while it was 36, and then become 50, and now you can go up to 300 bases. But you can see this spectrum. Once the increase to certain level, like 50 base, actually the mapability saturated. So you are not going to get a further coverage if you got a too much longer sequence. I mean, if you got way long sequence, it will cover it. But with the short range spectrum from 20 to 100, it's about 80% of the genome, human genome, is mappable. Well, some people will say, oh, that is contradict what you just said. 52% of human genome are repetitive, right? Why 80% is mappable? But keep in mind that those repetitive elements, they are not like one after another. So they are like here, 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 here. So a lot of them has the intersection border with the unique part of the genome, right? And so if one sequencing rate, one side covers into the unique side, the other side covers into the repetitive elements, although they are tapping into the repetitive elements, you can still map a dramatic portion of the repetitive elements. It's just the big repeats, you cannot really do anything about that inside. Any questions so far? Okay. So mapability is actually a big issue initially. I mean, people don't know how that will influence the performance of our analysis. So a lot of people did some previous work, as you can see. They look at different lengths of the sequences, and they will generate a mapability track. So I mean, you can define the mapability in different fashions. There are several ways for the alignment, but you can see this is what happens, right? So this is actually in the UCSC genome browser. There's a track called the mapability and alignability track. So you can see that for this part of the genome, and then with 24 nucleotide, and the average most of can be mapped, but there is a gap in the middle. And then with the increase to 100 nucleotide, so you can see the mapability increase dramatically. So this gap starts to becoming being filled. Okay, but keep in mind it's still about 80% of the genome is not mappable with 100 base nucleotide sequences. We need to be careful about this because sometimes that can give us a false indication about how we read through the data. And this is why that I'm always very favor of those tracks. I want to look at the data, not just some of the software generate Excel files, say, here are your variants, take from there, right? I want to go back, I mean, especially the variants that is important, I want to go back to the track to see whether my reads support that. Is that anything potentially wrong with the technology or the way that we do the analysis? So here is the issue that we need to be careful about. So one identified genetic variants, this is a read, and it could be, so there is so you can see these two elements, they are almost identical, okay? And except for there is one nucleotide difference, okay? So you got one sequencing race, and then if it has been mapped correctly, we know there are 2 homozygous variants in these regions. But if your alignment, because of this is more repetitive elements, it will be mapped wrongly to here, and your conclusion maybe this is a heterozygous variance. And actually, we had seen this before, and the only reason we know that was we were working on a male DNA sequencing data on the X chromosome, and apparently it cannot have heterozygous variants. And because the male only have 1 copy of the X chromosome. And then I saw this wonderful heterozygous variance in the male genome on X chromosome. And then we start to make a story of it. I said, wait a minute, okay? This doesn't sound right. And then we come back, we say, oh, there's a pseudo gene on the X chromosome. It's very similar to this, but because of the other variabilities in that region, that reads was a couple of reads were wrongly mapped to the other region. So sometimes it can be very, very dangerous. So we always need to be concerned about that. Okay, so be extra cautious after identifying variants. And then one we use this. Another one is when we look at the quantitative measurements when comparing 2 or more genetic regions. For example, if we work on the RNA sequencing data, so you can think of there are 2 genes and their expression levels are like that. But if there is another paralog, like this one, very similar to the red one, this gene also expressed, and some of these will be wrongfully mapped to here, and then you may have a wrong conclusion on this. So how to fix this problem, I really don't know, but we need to be cautious. Don't always trust what you see from your data. And the similar thing is when we look at the copy number variations, and you may see something like this, and there's no copy number variations, 1 copy of blue, 1 copy of red. But if you have a paralog of this region, if some of these rays are being mapped to here incorrectly, and then you may conclude that this red part has a copy number variation. So those are the things that mappability issues can really throw us under the bus for some of the studies. And another thing which we can do something about that is we mentioned capture technology, right? So when we do the whole genome sequencing, when we cannot do it, we will do the capture. And if you do whole exome sequencing, normally the microarray manufacturers will already, or whatever the adjuvant, they will take care of it. But if you want to target on the gene or region you are interested in, you need to design these target regions. And then, so the way that we do that, normally for this region we're targeting, we need to design a lot of probes to target these regions, and then this will hybridize to our DNA and pull it down, right? So if we are looking for PCR-based capture regions, and within the target regions, for this, we really need to worry about within these target regions, whether there are repetitive regions. So some part here is similar to some part over there, and we might have an issue on our analysis. But when we design, most of the target assays actually on the hybridization base, like a capture array or capture in solution. And we not only need to worry about whether this probe can be similar to somewhere within this region, but also somewhere outside of region. Because if that happens, if there's another region here is similar to here, and then you design a probe to target that, and that may also grab the sequence in this region back there, and if there is some variance that is, differences between here and here, you may wrongly, incorrectly conclude that there is a genetic variance there. So we need to be very careful in terms of the interpretation of the data. Okay? So those are something that we need to think about. And the mapability is determined by multiple factors. Actually, we know that the length of your sequencing rates will be important factor to consider. Another one is it may also be related to different alignment algorithms. Some of them are more sensitive than others. And so it's determined by the alignment algorithm, the regions where mapping occurred, and the biochemistry assays that are being used. So regions can be slightly different just because of the ability to deal with the CG or AT and then the tolerance. But these are just something that we need to be concerned about and keep in mind. Not too much you can do about a lot of things. Okay, so now I'm going to switch the gear a little bit. It's still related to mapability. Talk about a technology we didn't discuss last time in greater detail. We did mention that. Okay, so I stole some better slides now, so hopefully I can explain this a little better this time. So this is the 10X instrument that we just recently acquired in our center. And one of the capability, in addition to single-cell RNA sequencing, is to do the so-called linked read sequencing or long-range DNA sequencing. So here's how it's being done. Okay, so basically you can see that this is under the microscope. This is how it works. There's a bunch of gels. Okay, each gel carries a barcode. I mean, a lot of our oligos we had the barcode. Okay, don't worry about that. So you can see that this is the gel is pushing to this direction, and then this is your input DNA. This channel is your input DNA. Okay, and this input DNA normally will need to have this high molecular weight molecules. Okay, so what that really means, if you do the DNA sequencing, you just use a regular kit to extract the DNA from the blood, for example. And then if you run this on bioanalyzer, you will see your DNA fragments ranges from 300 base all the way to a couple of KB, right? And sometimes you will have to do the DNA fragmentation to bring the size down to about 600, 500 base pair-ish. That is not for this assay. For this assay, in the DNA extraction side, you need to have a very, very long molecular weight DNA, meaning that we need to have the DNA that by average 50,000 nucleotide long. So it cannot be break very often. So they show us during the training that 2 gel pictures, and they just constantly threaten us, be very gentle to the DNA and very, very gentle. And they show us 2 pictures. One is the DNA being perfectly handled. The other one is the same DNA being perfectly handled but dropped on the ground. And then they pick it up, nothing else, run on the gel so we can see that a band shaped. So those DNA are that fragile. So you need to be very, very happy. But anyway, so those are not the regular DNAs, but these are actually a much longer DNA, like a 50,000 base to 100,000 base DNA molecules. So once this is being pushed into, so you can see that at one step, your gel here is going to be attached to one or a few DNA molecules, and then this will be further pushed into oil. So this is a partitional oil. So we know water and oil, they are not the best friend. They don't go together. So you see that these will come into the bubbles, okay? Which means this gel and this DNA, high molecular weight DNA, will be trapped into this same bubble, okay? All the reaction, follow-up reaction will be happening in this bubble. So meaning this DNA will not be interacting with that. Okay, so you can see this is the partitioning part. Does that make sense? Okay, you have the gel, you have the high molecular weight DNA, you push that into oil and they form this droplet. Okay, so I'm not going to go into those very much detail on the technology side, but what you can see here is you push all these gels in, and then you collect those. These are called the gems now, okay? So they have the gel and they have the high molecular weight DNA. So you can see this, if you zoom out this, and this will have a very, very big gel, and then they have a lot of oligos, which has the barcode, the same barcode, and then you can see there's multiple DNA molecules in this. Multiple, it can be 5 to 10-ish, so if you control the reaction, it will be trapped in this way. Okay, so here's the trick why we're doing all these troubles. So now you got this DNA molecule. Okay, this DNA molecule is like a 50,000 base. Okay, so now you have this is part of the oligo on the bead. Okay, so they have this random primer, random sequence to do the random primer, right? Random priming. And then every single one attached to this little yellow tag, which is the barcode for this particular gel bead, okay, for this one. And then for different one, they will have a different short sequences, okay? So this is the way that you will separate, you say, okay, this molecule was generated in this gel, or this molecule is generated in this gel. So by analyzing the barcode part of it, okay? So now, once you do that, your DNA will be amplified, but with the barcode attached to it. So now I can pull everything back together, and then I do the sequencing. After I sequence, by reading the barcode, I will know which gel come from. And so I will know that a bunch of DNA molecules coming from the same initial molecule. Does that make sense? Okay? So that is how the technology works. So you can see this is another picture. It shows that this is gel, and it has oligos, which is, you can see those are random primers together with this green gel. So there's a green bar here as the barcode. And then you will do, this is your original 50 kb molecule, and then you will do the PCR for this. And then by random, a bunch of them a proportion of them will be put down and selected to do the whole genome sequencing. Okay, so you can see that each gem contains about 500,000 nucleotides, about 1 out of 6,000 of the genome. And so if your average molecular weight is 50,000 base, each fragment, and then each gem, you will have 10 high molecular weight DNA being trapped. They will share the exactly same barcode. Okay, so if your average is 30x sequencing for the genome, and by the end of the day, and for each of these molecule, your coverage is about 20% of the DNA molecule will be covered. Does that make sense? The last part? So we know that when we do the whole genome sequencing, and we're going to target for every nucleotide, we'll have 30x coverage. That's by average, right? And we know we have over 1 million gels as well during this reaction. So in that case, for each individual molecule, okay, we will not cover 30 times. We will only randomly select a proportion of them being covered. So that proportion is about 0.2x coverage. So for each molecule, there will be about 20% of the site will be covered by your sequencing. But if you look at whole genome sequencing, each position will be covered 30 times because we have 1 million of those gels. Okay? So this is basically effectively the long linked rate sequencing. It's the same as the previous one, except for it has this permutation step that you can barcode each individual molecule. This gives us a lot of advantage. Okay, so I'm going to talk about to increase the mappability part, but next time we'll talk about the application of this to help us to identify the haplotype of the DNA and then also to help us identify the structure variations. So those are incredibly helpful. Okay, so let's say in terms of mappability part, so here is how that happens. There's two regions, genomic regions. They're similar to each other, and the blue, and then this dark blue. It's more like a black. It's on my side, it's dark blue. And they are parallels. They are very similar. The sequencers are similar to each other. And then both these two regions are kind of adjacent to a unique region. So this green and the yellow is not similar. So if we do the regular sequencing, if we do the mapping, we know those green and the yellow read will be mapped pretty easily because they are unique regions. But for the remaining ones, they will also be aligned, but in some of the cases, they may be aligned to the wrong position because these two genomic regions are too similar to each other. That make sense? Okay, so that will cause a lot of problem because you incorrectly call some of the genetic variants just because of that. Now with the linked read sequencing, and the first round is still very simple, right? So you can see that there are reads being uniquely mapped to these unique mappable regions. Now we're going back to see this attachment, which is the barcode. So you're seeing that this molecule has the barcode of yellow. So that means the yellow read will be more close to these regions. And the same thing is the purple read will be more close to that regions. In that case, if you do the alignment properly, you will know that those regions will be mapped. And taking the advantage of this entire molecule is being barcoded. Any question? Yes? 50,000, and then you have to break it. so yes. So you can see that in this part, when we do the PCR, and these are random primers. Okay, these random primers, and then we prime here, and then do the sequence, synthesize, right, amplification. And then this will not cover the entire, all the way towards the end of the model. It depends on your annealing time or, extension time or whatever.
So each round would have the same barcode?
Yeah, so each of these elements will have the same barcodes. Yeah, you're right. The initial molecular weight is very high. It's a 50,000 bases. But once you get to the library stage, each DNA fragment is not that high. The initial one is only as a template. Okay? Any questions so far? This is cool, huh? This is really cool technology. And then I went to the internet and trying to find out some of the evidence this really helped. In theory, it will. And then I found out a poster that is presented in American Society of Human Genetics meeting in 2016. And so you can see their conclusion here is 322 protein coding genes in regions covered by linked reads in the majority of our sample, but not in the standard library. So you can see in these regions with the linked reads 10x data, it has been covered very well, just because of those mappability issues, we link them together. And then in the standard sequencing, whole genome sequencing, a big chunk of those regions are completely not mappable. Okay, we know this is so important for a lot of disease, right? And many of the important genes are actually A genes, and there are many things are in the very much repetitive regions. Okay. Any questions? Okay, let me drink some water. I learned all this week, so if you didn't get it, don't feel too bad. Okay, so the next topic I want to get into is refined alignment. And the first question is what we are talking about. We already did alignment. We got the sequencing rates. We mapped to a certain part of the genome. What else do we need to deal with? Okay, so let's revisit our initial sequence alignment. We spent quite a lot of time in the third lecture talking about that. So whatever we say, if you look at the goal of the initial alignment, here's the goal. The number of mismatched bases will be minimized across one single sequencing rate. Okay, so if we got this one, this is a reference, this is one sequencing rate. If we map to here, and then our goal is to minimize the mismatches, okay, using all different approaches. Well, now refined alignment is within a genomic region here, and we know that we are not going to have only one sequencing rate. We'll have many sequencing rates stacking together. And our goal here is the number of mismatched bases minimized across all the sequencing rates. So that is the goal, not only based on the one rate, okay? So just to keep in mind of this statement, so I will show you an example to make sense of it, okay? What went wrong for the initial alignment? A large percentage of regions requiring local realignment are due to the presence of an insertion or deletion in the individual's genome with respect to the reference genome. Okay, so let's go through this figure. This is a complicated one, so you have to follow me like a BW, like the Borel-Weber transformation, right? You have to follow me on this. So what went wrong? So you can see that here is an example. This line here is, I'll use this. So this line here is the reference genome, okay? And this line here is our sample, the donor genome, okay? The major difference between these two is there is a 4-base insertion of the donor genome compared to the reference genome. We got it? Okay. There's one thing I want to point out. The reference genome is visible, and we don't have a sample genome. We don't have the donor genome, right? This is just putting here to show you that that is what the answer is. If we already know the answer, there's no point to sequence these individuals, right? So we don't know the second line, this sequence. Just sometimes make it invisible. Okay, so now let's go to individual rate. We did the whole genome sequencing or whatever on this particular individual. And then we got 6 sequencing rates across this region. Let's look at the first sequencing rate. Okay, so there are 2 possibilities we can do the alignment. Either we can do it correctly, which is not this one. So you can see this is a AAATAA. And it should be AAATAA. So this one should be perfectly aligned to the sample genome if we know the sequence of the sample genome, which we don't, right? So the software itself, the alignment algorithm itself, has to do the best job they can. And if they do in this way, in the correct way, so you can see this 1, 2, 3, 4 nucleotide difference compared to the reference genome. If I'm aligning in this way, there is only 1 nucleotide difference from T to A change. Does that make sense? Let's look at the second read, okay? And everything looks good, and then it's become AAAT, AATT. So if you align this correctly, it becomes AAAT, AATT. Actually, this is a perfect match to the donor genome if we have it. Unfortunately, we don't. So when the algorithm does the alignment, it can only align back to the reference genome. So this way, you will have 2 nucleotide differences. If you align those correctly, you will have 4 nucleotide difference compared to the reference genome. Does that make sense? If you go through this here, this one is kind of in the middle, and if it cannot make mistake. If it make mistake, everything else afterwards will be wrong, right? So you can see this is one of them has been aligned correctly, and then but consider on the donor on the reference genome there will be 4 base differences. Okay, so this one is on this side, so you can see 2 base differences are not being aligned correctly. If you do correctly, it should be no difference compared to the donor genome. And then the same thing here, the same thing here. Have you got the point here? So if we do the initial alignment, we can only vary about 1 single nucleotide at a time, okay? Because the donor genome is not visible. We don't have it, okay? So that's why for this region, the end result we got is after you do the variant calling or whatever, you see it in the genome browser, you will see there is a frequently, there are some of them are having a small insertion deletions, but most of them don't. And then there is a lot of sporadic variabilities around those regions. So those are the symptoms of this region that I probably need to do the refined alignment. So let's see. So such alignment artifacts result in many base mismatching the reference near the misalignment, which is mistaken. Sometimes it can be mistaken by SNP as well. Not only you are not able to identify this indel, but you are mistakenly say, okay, here's the SNP, here's the SNP. Sometimes that could happen. That's again the reason if I'm asking you in the exam, what's the reason we have to do the refined alignment? And what's wrong with the initial alignment? Actually, the answer for that is the initial mapping treats each individual read independently, and then coming back to the previous slide, because it tries to minimize the mismatches for 1 single nucleotide read. So even when some of the reads are correctly mapped with the indels, reads covering the indel near just the start and end of are often misaligned. So we should talk about this part. Okay? So that's why we have to go for local refined alignment or local realignment. Serves to transform regions with misalignments due to the indels into clean reads containing a consensus indel suitable for standard variant discovery approaches. Okay. To do that, there are 2 approaches, so I'm going to lead you through how to do it. Okay. There is a code, a software to do it. You don't have to do it. But you don't have to write the code to do this, but here is how the software identifies those regions and then further fix it. So there are 2 steps. The first step is to determine the suspicious intervals which are likely to be in need of refined alignment. Okay, so you have to identify those regions, and we need to do this. The step 2 is to fix it. Okay, so first of all, how to detect those regions. So we have mentioned briefly, but here is what people do is if 1 or more read contain an indel in them, okay, like the example we show, 1 read have an indel in that, and are aligned correctly, one would want to make sure that the other indel containing reads in the pile up are aligned correctly. So this is if some of the region has some indels being identified, and that is a region that we probably want to go back to the refined alignment. The second one is if no indels are identified in the initial alignment, and you want to search for the cluster of SNP calls, which means after you do the SNP calling, you found there are so many SNPs that in a very closely related regions, that is a suspicion that that region needs to do the refined alignment. And then if you haven't done the base call, and then you want to detect a cluster of loci with high entropy, meaning there's a lot of disagreement of different reads in those regions. So those are the strategy that the software does to identify the regions that needs to be going through refined alignment. And also, people also will put in known indel regions in the DB SNP into those. So those are the strategies to identify that. Okay, so the second step here is to do it. Again, this is two steps. The first one is construct all possible haplotype by integrating reference genome, known gaps, and the de novo gaps. Okay, which means, and then the second one is to do the gapless alignment. So we'll go through this. Okay, so the first step here, there's two steps. The first step is construct all possible haplotypes by integrating the known genome, known gap, and the de novo gap. So what this really means. Okay, so you can see that this is your reference genome. Okay, this is a potential haplotype. This is a potential reference genome that we still will consider as one of the options. So the second one here is going to be a different haplotype that will be evolved from your data, which means if I have one sequencing read, identify a small indel, and then I will put this indel into this region and make a new reference genome in this region. That makes sense? So that is the second haplotype. So if you have multiple indels being identified in this region, you will increase or list. But this is two options so far. The original reference genome with the de novo reference genome that were being derived based on one or two or a few of the indels already being identified in your initial rates. Okay? Does that make sense? So that's the first step. The second step is we want to pick which of these two are likely to be the important one, the correct ones. Okay? So the way to do that is to use this equation, okay, which I'm not going to go through. But the point here is you actually, given each individual haplotype, you want to calculate the likelihood of this haplotype based on all the reads in this region. Okay? And then, so this is the number of the sequencing reads, which is J. Okay? And this is the likelihood of if this one haplotype is correct, and then what is the likelihood of observing that particular read. And this one, it goes to here, and then which is the number of mismatches here, and then if this is the correct haplotype, and what are the opportunities I'm making mistake here, right? So there's a number of mismatches, and for each individual mismatches, you will give a score for the match, and you will have a good score, which is 1 minus a small error possibility of errors, and or if it's not a match, it will be this. So I'm not going to test you on this. You can think, if you are more math-oriented students, you can go back to think about this, and it's actually not that complicated. But the point is, now you have two possible haplotypes. You want to select which one is right. You want to align all the remaining reads here to both of these two haplotypes. We're using the so-called gapless alignment algorithm. So you are not allowing any insertion deletions for the alignment. When you align this to here and to here, okay? Because it's not going to happen anymore. So in that case, you will see that from these two, what are the scores? What are the possibilities that you are doing this correctly? And by calculating the likelihood of each individual two of the haplotypes. That make sense? Okay? Roughly makes sense? Okay? Vaguely makes sense? Okay, go through your lecture notes and or the video recording, and then you try to make sense. So you see the haplotype HI that minimize this is selected as the best alternative haplotype and assign all the reads either to H0 for this one or to the selected bias haplotype. And then the next one is you are going to calculate a likelihood ratio to see that if I'm picking this haplotype, how much more gain I will get comparing to using the initial haplotype. Okay, so this part is more mathematics oriented, which is not required here, but I want you to get the point that to do this is a two-step approach. The first one is construct all the possible haplotypes by integrating all the information you have so far, and then align all your reads into both two haplotypes to see whether using the new haplotype will help you dramatically to increase the overall performance of the alignment. Yes?
The new haplotype which is created from the reads itself.
Mm-hmm. From possibly one read. And a proportion of the read that has that haplotype, has that insertion detected. So the point is I want to see whether it actually fits the others better, right? So and I never had the opportunity to map the other reads which were wrong or incorrectly aligned to this new haplotype. If I do that, whether that will make more reads being aligned. That's the initial point. Is for the initial alignment, we can only take care of one read at a time, and then to map to the reference genome. Now we can take all of them at a time, and then to minimize the overall mismatches. That's the whole point. Okay, and in the real situation in the software, what it does is the read are realigned if the log odds ratio of the two haplotype model is better than the single reference haplotype by at least 5 log units. So which means that if my new haplotype is doing better than using both two haplotypes is better than using only the reference one by a little bit, I'm still not picking the new haplotype. Okay, I need to have this gain needs to be substantial. It needs to be 5 times log units. That is a lot better in terms of the mapping probability. And then so here is the way that you do the calculation. Okay? Yes.
Like the other example where we had more mismatches.
Yeah.
Now, in these cases, you're just trying to put in like 2 or 1 or 2 in one.
So if we do, let's say, we go back to the original example here, right? This one. So now that this particular read doesn't change at all, right? So the middle read number 3 doesn't change at all, right? But for this one, so you can see that when I map to this, and I found there's a mismatch, and then I will get a score penalty, right? Because I got a mismatch. But when I align to this one, and then everything is perfect, right? Yeah.
How do you know when I'm going to this one? Because this one has 4 mutations, and your new reference, you might want to be with your mutations.
My new reference is here, right? It's this one. So this will be my haplotype 0. This is the haplotype I, right? The new haplotype. So if I align this race to here, there will be 1 mutation. If I align this to here, there will be 0 mutation.
Yeah, that's true. But like when you're creating the reference genome itself, here you're trying to put in 4 different mutations there. But you said maybe 1 or 2, you need to add 1 or 2 in those into the new.
Well, you're right. If I'm only looking at read number 1, and I probably won't pick this, right? So now I'm looking at read number 2 as well, right? So you say, oh, map to here is perfect. And then if I have the ability to fix all of this other reads, and then your overall gain of adding in this new haplotype will be more dramatic. So that's exactly the initial point, that initial alignment only take care of 1 read at a time. So you cannot make that decision. But when you have take a more democratic process, like all the reads to vote for which haplotype is my best one. But if I have to throw out the current government, and then I will have to have substantial vote to do that. So that's that's how the overall thinking process of this. That make sense? Okay. So again, for this part, this whole section, I think again, the math is not needed, but I do want you to understand that a few information points that may be in the exam. So before we get into that, let's just see the performance. So here is the example in the initial paper. So you can see in this region, this is before they have the refined alignment. And then you can see there is 2 SNPs are being identified. Okay? They are being identified incorrectly, actually. And then, but there is some deletion rates detected. After fixing it, this is actually a more realistic one. So you can see half of the rates actually has a deletion, half doesn't have the deletion, and there is no other SNPs in this region. Okay? That makes sense? This is another example. So you can see in this region, it's pretty colorful, right? So you had this T, this T is always there. Don't worry about that. But here you got a deletion, and then you got a couple of variants. But after you do the refined alignment, you can see this part become much cleaner. And actually, all this part is just because of this one homozygous, almost homozygous imbalance in here. And so this, if you don't do the refined alignment, I'm pretty sure that there is 2 false positive variants are being caught using the original alignment algorithm. Yeah.
What software can you use?
It's GATK. GATK, yeah. It's GATK. So the reference is here. So for this lecture and the next lecture and most of the part, I'm going through GATK package because this is becoming a standard for the DNA sequencing variant calling analysis. Okay. So the key information point I want you to understand is the first one is why this is needed, which again, whether it's to minimize one read, a mismatch for one read or for multiple reads. And I also want you to know this, to do this, there are 2 steps process. The first one is to identify suspicious regions. And if some of the reads are 1 or more, identify indel, that is a suspicious region. Or if there's many variants or inconsistency in a very small restricted region in the genome, that is a suspicious region as well. And some people also put the dbSNP index into that as well. So the second step is going to do the refined alignment, and this also have 2 steps. Well, it's 3. Okay, 3 steps. So the first one is you want to enumerate all the potential haplotypes based on initial alignment. And the second one is to conduct gapless alignment on all the haplotypes. Gapless, this is the key for here. And then the last one is to calculate the likelihood for each individual haplotype and whether that will give you significant gains. Okay? So these are the information points I want you to understand about the refined alignment. In addition to the homework that you are going to identify some, do that using the supercomputer services. Okay. There is something that I want to think about. I feel this we need to use this approach with a caution. Because we need to understand what are the major assumptions here. This algorithm operates on all reads across all individuals simultaneously, which ensures consistency in the inferred haplotype among all the individual reads. That is a good thing, but it works on if I have 2 alleles. And this is germline mutations because you need to have a substantial number of the reads that support this indel, this new haplotype, right? At least half if it's a germline mutation, right? But if you're working on a tumor sample, this assumption may not hold because if this is the indel from the tumor sample, you may have only a small proportion of those sequencing rates that are coming from that cell. And then you may have only maybe 10% or 5% of your reads carry that indel. In that case, in theory, this can still help you to fix it, but the way that they decide whether the second haplotype being selected is you have to have substantial gain across using only the original reference. So if only a very small proportion of the reads has that legitimate indel in that, you may not have enough gain so that the software will not pick that haplotype. And it needs to have significant improvement to replace the initial alignment. May not work for pooled sequencing or RNA sequencing either. And just to go back to think why that's the case. All right. Any questions about the refined alignment? Yes.
Is the requirement for refined alignment increased with read length, or what's that? How does that work? Is increasing read length is required, or does the requirement reduce?
No. So far that, I mean, most of the standard assay to do the DNA sequencing is to use the 2 by 150, which means 150 paired end reads. Sometimes 100, but most of them 150 paired end reads. And this is, once you do the DNA sequence, genomic DNA sequencing, this is a very standard process. You cannot skip to do the refined alignment. So it's not have anything to do with the read length anymore, and you will need to do that. Other questions? Okay. All right, so next, we're going to talk about the quality and the quality recalibration. And this is more refers at the base quality recalibration and not on the variant yet. So we'll talk about the other recalibration next lecture. Okay? So let's, so basically it will be this part. So the question here is when Illumina gave us the sequence, and then for every single nucleotide, and we will have a Q value, right? Remember we talk about Fred scores and things like that. But the question is why should we trust what they gave us, right? That's not something we derive, that's something they tell us. Just a little bit of recap from the previous lecture about the quality evaluation. So you can see that we will do this Q value calculation, which is minus 10 times log 10p. And then, so for the probability, if this is, you have 1% of the chance to make mistake, your Q value is 20, right? If you have 0.1% chance to make mistake with Q value is 30. Meaning the larger the Q value is, the more accurate your sequences are. So these base quality, they are reported by the sequencer, but are they really accurate? Without a positive control, which means you know exactly what you are sequencing, in most of cases we don't, it's very hard to evaluate, right? But what people did is, for this one, again, this is also part of the GATK package, and we just go to the theory part of it to see why it's necessary. So people did some analysis. So you can see this, there are 4 different platforms that people look at. Illumina, which is the earlier version, GA version, genome analyzer, and the Roche 454, it's no longer in the market. Life Solid, no longer in the market, and Illumina HiSeq 2000. It was such a cutting-edge technology at the time, the first time I teach this course. Illumina 2000, and then after that we 2500, and we had our first one, which is 4000. Now we have NovaSeq 6000, and nobody is using HiSeq 2000 anymore, right? But if you look at here, what this figure tells you is X is the reported quality, okay? And Y here is the so-called empirical quality. And you can think of this is the true quality, the real quality. And X here is the reported quality. Y here is the real quality. We'll go through how they estimate the real quality, which may not be perfectly fair. But what you can see here is that there is a significant discrepancy here. And the purple ones is before we do the refined, before we do the quality recalibration. So you can see the reported quality is constantly higher than the real quality. Okay, so this happens in Illumina earlier version. Let's forget about this. They're not being used anymore. And then for the HiSeq 2000, it's also have the same issue. So which means they are not very faithful. They give us the better score than they should give us. Okay. So before we get into what that caused that and how that will be fixed, and I want to just briefly talk about how the Y is being generated. X is easy. X is reported quality, right? Y is empirical quality. So the way they do that is they calculate mismatch at the loci without known variance. So basically, for all the sequencing rates, they will do the calculation. They say, okay, if there is a reference genome in the dbSNP, there is no indication there is a SNP in that. And then if there is a mismatch on my sequencing read, I will consider that as a incorrectly identified variant. Okay? Not fair. So in many cases that may not hold true. So this empirical quality may be a little bit too strong assumption, but still, and you can see that there is a substantial discrepancy between these two numbers. And they also further evaluated what is associated with these qualities. And the first one they look at is the machine cycles. So meaning that the so-called machine cycle is the position on the nucleotide. Whether it's the first read, the second, the third, the fourth. Now if we do the 150 base read, and that will be machine cycle up to 150 base. So you can see that for the Illumina technology, it's pretty constantly overestimate their quality. So this Y here is the empirical minus reported quality. So if it's a 0, that means the reported quality is accurate. If it's a minus, it's a negative number, that means reported quality is actually better than the real ones. Okay, so you can see they are all going below 0. That means they are across the board at all different positions. They're kind of have over-reporting issues. And then, but for Roche, for 454, it's a slightly different pattern. For solid, it's something like that. Some of the base they overestimated, and others they are underestimated. And then you can see for Illumina here, and they are all pretty consistently overestimated their quality. So that is the machine cycle. The second one they look at is whether there is any association with D-nucleotides. So basically, they look at all the possible D16 type of dinucleotides, dinucleotides, AA, ACA, GAT, or whatever. And so you can see that at some of the sites, some of the dinucleotides, the reporting was accurate. And some of them, they are not, especially for CG. Those are important ones. Those are potential methylation sites, right? And this is over reporting. And actually, this happens in all different platforms as well. And actually, they characterize much more than this. I'm just giving you some of the examples. So these higher than empirical or over-reported quality sometimes will provide a false confidence to your data, right? So if in reality it should be Q20, but you reported 25, and then you give me a wrong confidence, and that may be a problem. So this is a consistent across all the different platforms. They are all having the over-reporting problems. So what is causing this problem? And I mentioned earlier, and there are three covariates we can consider. Number one is what is the number they reported. Okay. So the second one is the probability of and the position within the read. So meaning that the machine cycles. And the third one is the preceding and the current nucleotide, the dinucleotides. So this is what I mentioned. So all these three will have some indication. But those are the possibility of this recalibration may be overstating the problem a little bit as well. So the question is, if they over-reported, how we correct it? And next lecture, when we talk about the variant calling, I will tell you many variant calling algorithms will require accurate reporting for the base quality. So correcting this is very, very important. So these covariates, reported quality, machine cycle, and dinucleotides, those are the three factors I showed you, are subsequently applied through a piecewise tabular correction to recalibrate the quality score of all reads in the BAM file. Okay, forget about that. Those or take a couple of hours to think it through what that really means. This is what that really means, which converted to the equations, which is not intuitive. So let me give you an intuitive version of it. Okay, so here is your reported quality. And what they did is they will adjust this reported quality. Okay, so the way they do that, they will give a delta QR, so meaning that if your reported quality is a number, and then they will calculate all the reported, so all the Q30 base, and they will see what are the real quality of that. And then there will be a difference on that, and they will record that. So this is only specific to the reported quality. And then there will be something related error for all reads with reported quality as R, as well as the nucleotide positions. So if there is a difference on that, you will make a further correction. And the third one is to look at the dinucleotides, and then to see whether we need to do the further correction. And then after this three level of correction, and this is going to be your recalibrated quality. So let me give you a real example of this. I mean, not a real example I made up. And for one specific base, the reported quality is 30. That tells you about 0.1% of the chance this will make mistake. And then in the whole experiment, the average imperative quality for all the base, the reported quality is 30, is 25. Okay, so they will take all the base, say the reported quality is 30, and they will calculate it. Actually, the empirical quality of that is 25. It shouldn't be 30. So then the delta QR, the first correction, is -5. That makes sense, right? You're over-reporting. And then the second one is the base locates in the fifth location, because that one specific site is the fifth location in the nucleotide. In the read, the average imperative quality for all the base with reported quality as Q30 and at the position number 5 is 27. And then your delta QRC, this will be positive too, because by 25, actually, because it's only in the beginning of the reads, you're kind of not too much over-reporting, and then you have gain a little bit credit of Q is equal to 2. And then the next one is the dinucleotide of the sequence result is CG. We know this is a hard one. And then we know the average empirical quality for all the base with reported quality as R30, and the dinucleotide as CG is 23. Now it should be 25 or 23. So you have an additional penalty of -2 for this. Okay. So by working through all this, your final quality, recalibrate quality, will be 30 minus 5 plus 2 minus 2 is equal to 25. And that is converted data to 0.3% of the chance that you are making a mistake, not 0.1%. Does that make sense? Okay? Sort of? Okay? All of the stuff that we talk about today is kind of, some of them you cannot get it immediately. We'll think a minute. You need a minute to think about that. So let's briefly look at the performance after the recalibration, which is the blue dots. So you can see we don't need to use any word, but you can see that this goes to 45-degree line, meaning that after recalibration, the recalibrated quality is similar to empirical quality. And for this tool, and they are all going to about 0 points, which means there's no discrepancy between the two. Okay? And the example I gave you is to use 3 covariates. Okay? The reported quality, the position in the read, and then the dinucleotides. In reality, there are many of them, and they are all being considered in the GATK package as well. So you just need to go ahead to use it. Okay? So next lecture will be similar as this one, still heavy. And we should talk about the variant identification, variant quality recalibration, and the structure variation identification. And then after that, I will take a break. And then that will be Dr. Dongming Lai talk about the statistical genetics, and then will be still me, and then I think my lecture is almost all gone, and only maybe 2 or 3 more lectures left after this. Oh, by the way, after this, we will not too far away from midterm, okay? Just to remind you. Okay? All right, if no other questions, everybody have a good weekend.